Dear students, let us continue our lesson. Cell, the unit of life. Now we shall study the prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. PPLO means pneumonia-like organisms. These are the examples for prokaryotic cells. Bacteria, the primitive organisms. Blue-green algae, now we call cyanobacteria. Mycoplasma, then PPLO means pneumonia-like organisms. These organisms, that is PPLO, cause respiratory disease in fowls. Okay. These are very primitive organisms. They are smaller and multiply rapidly than the eukaryotic cells. Even they may vary in shape and size. That is, prokaryotic cells vary in shape and size. Say, bacteria. Bacteria are classified into different types based on some criteria. For example, bacteria classified into different types based on their shape. So, based on the shape, classified into 4 types of bacteria: Bacillus, Coccus, Vibrio, and Spirillum. Bacillus bacteria occur as rod-shaped cells. The bacteria with rod-shaped cells are called bacilli. Bacillus is singular. Similarly, the spherical-shaped bacteria are called coccus. Cocci, plural. Some bacteria appear comma-shaped. So those are called Vibrio. And some bacteria appear as spiral-shaped structures. They are called spiruli or spirulum. So, based on shape, rod shape, spherical shape, comma, and spiral shape, bacteria are classified into 4 different types. Okay. Now, let us see the organization of the prokaryotic cell. ಹಂಗಂದ್ರೆ ಏನಪ್ಪ? ಸೆಲ್ ಯಾವ ಥರ ಇದೆ? ಪ್ರೊಕ್ಯಾರಿಯೋಟಿಕ್ ಸೆಲ್ ಯಾವ ಥರ ಇದೆ ಅಂತ ನೋಡ್ತಕ್ಕಂಥದ್ದು ನಾವು. The organization of the prokaryotic cell is fundamentally similar. Even though prokaryotes exhibit different shapes and functions. All the prokaryotes have cell wall. Means the outermost layer is the cell wall. Next to the cell wall, we find cell membrane. The cell membrane includes or inside the cell membrane, the fluid matrix is present. The fluid matrix. It is called cytoplasm. It means the outermost layer is cell wall. Inside the cell wall, cell membrane is present. Inside the cell membrane, the fluid matrix, cytoplasm is present. Okay. In the cytoplasm, the nucleus is present. There is no well-organized nucleus. Well-organized means the nucleus must contain nuclear membrane, nucleoli. Such nucleus is absent. The genetic material is naked. The genetic material means the chromatin is naked. Naked means it is not surrounded by nuclear membrane. In eukaryotes, we have nuclear membrane. This nucleus is called prokaryotic type of nucleus. A primitive type of nucleus. So this is we call genomic DNA. The chromosome is in circular shape. the bacterial DNA is circular. It is okay. It is called genophore. So very important point. The nucleus is prokaryotic type. Means the nuclear membrane is absent, represented by the circular DNA only. Okay. Now, if we examine carefully the prokaryotic cell, in addition to this DNA, that is circular DNA, we call genomic DNA. In the cytoplasm, smaller DNAs are present. The circular shape. These are circular shape. We call plasmid. Don't confuse. Prokaryotic nucleus è ಬೇರೆ, plasmid è ಬೇರೆ. So prokaryotic nucleus is the bacterial DNA, the plasmid. What is the meaning of plasmid? I will tell you. The plasmid is nothing but extra circular double-stranded self-replicating DNA. So such structures are called plasmids. Plasmid is nothing but extra DNA. Why we call extra DNA? Because the genomic DNA is different from plasmid DNA. It is circular in shape. It's not helical. Self-replicating. A plasmid can produce another plasmid. Okay. The number of plasmid also varies. So, ಇಲ್ಲಿ very important point to genomic DNA ಇದೆ, ಜೊತೆಗೆ plasmid DNA ಇದೆ. See the diagram of a typical eukaryotic cell and different types of bacteria. We discussed just now. See the diagrams of different types of bacteria. Okay. The plasmid DNA brings about a special, we call, unique character. Unique character to the unique character, that is what we call phenotypic characters to such bacteria. Say for example, one such character is resistance to antibiotics. So plasmid gives resistance to antibiotics. But if you examine the eukaryotic cells, in case of eukaryotic cells, the nucleus is eukaryotic type. That is, it is having nuclear membrane, and inside that nucleolus is there, nucleoplasm is there. You can see the eukaryotic cell. In addition to this, eukaryotes have cell organelles. But the prokaryotic cell does not contain cell organelles. So very important is the eukaryotes have eukaryotic type of nucleus. Prokaryotes have prokaryotic type of nucleus. Both of them have the ribosomes. Ribosomes ಎರಡು ಸೆಲ್ಲಲ್ಲೂ ಇರುತ್ತೆ. ಓಕೆ, prokaryotes ನಲ್ಲಿ cell membrane forms a special structure called mesosome, which is nothing but the infoldings of cell membrane. Okay, that's all about a prokaryotic cell. And we have discussed a brief account of eukaryotic cell also. Okay, now we shall discuss about cell envelope and its modifications. Most prokaryotic cells, means the bacterial cells, have chemically complex cell envelope. Cell envelope means the covering. The cell envelope or the covering consists of tightly bound 3-layered structure. The outermost glycocalyx, followed by the cell wall, and then we have plasma membrane. See, glycocalyx, cell wall, and the plasma membrane. Each layer perform different function. But they act together as single protective unit or layer. So, bacterial cell wall alli ಏನೇನಿದೆ? ಗ್ಲೈಕೋಕೈಲಿಕ್ಸ್ ಇದೆ, ಸೆಲ್ ವಾಲ್ ಇದೆ, ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇದೆ. All these 3 layers, they are the protective layer together. Okay. Now, next we shall see the classification of the bacteria. The bacteria can be classified into two groups on the basis of differences in the cell envelope. That is, when we stain the bacteria, okay, on the basis of differences in the cell envelopes, they are classified into two types. The bacterial cells, when we stain ಬಣ್ಣ ಅಂತ ಕರಿತೀವಿ, ಡೈ, that is Gram stain. Gram stain ಅನ್ನೋದು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸ್ಟೈನ್, ಡಿಸ್ಕವರ್ಡ್ ಬೈ ಗ್ರಾಮ್. ಅದಕ್ಕೋಸ್ಕರ ಇದನ್ನು ಗ್ರಾಮ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಗ್ರಾಮ್ ಸ್ಟೈನ್ ಅಂತ ಕರೀತಾರೆ. When the bacterial cells are stained with gram stain, some of the bacteria take stain and others do not take stain. The bacterial cells which take stain are called gram-positive bacteria. Gram-positive bacteria. ನೋಡಿ, ಅದು ಮಿಸ್ ಆಗಿದೆ ಇಲ್ಲಿ. Right, it is there. Gram-positive bacteria means the bacterial cell wall takes stain and appears blue color. Gram-negative bacteria, the cell wall do not take stain. This is how the bacterial cells are identified. Bacteria cell-ನ ಯಾವ ಥರ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತಾರೆ, ಯಾವ ಥರ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಾರೆ ಅಂತ ನಾವು ನೋಡಿದರೆ, ಸೊ ಗ್ರಾಮ್ ಸ್ಟೈನ್ ಅಂತಕ್ಕಂಥ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದೆ. ಇದನ್ನು ಸ್ಟೈನ್ ಮಾಡಿದಾಗ ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸ್ಟೈನ್ ತಗೊಳ್ತವೆ, ಅವನ್ನು ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್ ಅಂತ ಕರಿತೀವಿ. ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸ್ಟೈನ್ ತಗೊಳ್ಳಲ್ಲ, ಅವನ್ನು ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಂತ ಕರಿತೀವಿ. ಈ ರೀತಿಯಾಗಿ ಎರಡು ಥರ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ, ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಓಕೆ, ನೆಕ್ಸ್ಟ್, the glycocalyx differ in composition and thickness in different bacteria. It may be appear as loose sheet. Then it is called slime layer. If the glycocalyx is in the form of loose sheet, then called slime layer. If glycocalyx is thick layer and it is tough layer, then it is called capsule. there is a difference between slime layer and capsule. This actually determines the shape of the cell, and it supports the bacterium from bursting or collapsing also. That is glycocalyx. Okay. Next, ಏನಿದೆ glycocalyx ಆದಮೇಲೆ next to the glycocalyx, plasma membrane is present. Plasma membrane is selectively permeable membrane. And this membrane is structurally similar to the eukaryotes. Okay. Next, very important thing no, now. This plasma membrane in the bacterial cells form what is known as mesosome. The mesosome is nothing but the infoldings of the plasma membrane, and it forms the vesicle, tubule, and even lamellae. They help in cell wall formation. That is nothing but the function. It also helps in DNA replication and distribution to daughter cells. Mesosome also helps in respiration. In bacterial cells, there is no mitochondria. So mesosome takes the part of mitochondria. It helps in respiration, secretion, and increased surface area of plasma membrane, and enzyme content. Okay, that is, the function is cell wall formation, DNA replication and distribution to the daughter cells, respiration, secretion, then enzyme content. Okay. In blue-green algae, plasma membrane forms what is known as chromatophores. Chromatophore means a primitive type of plastids, you can say. Eukaryotic cells ನಾವು ನೋಡಿದರೆ ಪ್ಲಾಸ್ಟಿಡ್ಸ್ ಇರುತ್ತೆ. ಬಟ್ ಪ್ರೋಕ್ಯಾರಿಯೋಟ್ಸ್ನಲ್ಲಿ ಪ್ಲಾಸ್ಟಿಡ್ಸ್ ಇರೋದಿಲ್ಲ. ಪ್ರಿಮಿಟಿವ್ ಟೈಪ್ ಇರುತ್ತೆ, ಅದನ್ನು ನಾವು ಕ್ರೊಮ್ಯಾಟೋಫೋರ್ ಅಂತ ಕರಿತೀವಿ. ಕ್ರೊಮ್ಯಾಟೋಫೋರ್ಸ್ ಕಂಟೈನ್ blue-green pigment. Why we call blue-green algae? Because of the presence of a special pigment, cyanophycean pigment, blue pigment. Okay. Then the flagella may be present or absent. On the cell wall, flagella may be present or absent. If the flagella present, flagellum is made up of filament, hook and basal body. Okay. In addition to that, we find the pili and fimbriae occur as surface structures which helps in attachment to rocks in streams and also to the host tissues. But do not play any role in the mortality of the cell. That is pili and fimbriae. Okay. Then what are pili? Pili are elongated tubular structures with a special protein. Then what is the difference between pili and fimbriae? The fimbriae are bezel-like structures. They are bristle-like structures. Pili are tubular-like structures, elongated tube-like structures. Tubular means. Okay. Now, next you shall see the ribosomes. The ribosomes of prokaryotic cell are called 70S ribosomes. I told you the ribosomes are present in prokaryotic cells and also in eukaryotic cells. Okay. The main function of ribosome is protein synthesis, both in eukaryotes and in prokaryotes. If we examine the ribosomes in prokaryotes, as I told you, they are of 70S type. Whereas the ribosome of eukaryotic cells are 80S. This S refers to Swedberg unit. It means sedimentation coefficient. Sedimentation coefficient ಮೇಲೆ ಎರಡು ಥರ ಕ್ಲಾಸಿಫೈ ಮಾಡಿದ್ವಿ ರಿಬೋಸೋಮ್ಸ್ನ. ರಿಬೋಸೋಮ್ಸ್ ಆಫ್ 70S, ರಿಬೋಸೋಮ್ಸ್ ಆಫ್ 80S. Ribosomes of 70S are characteristics of prokaryotes. Okay. Now, in prokaryotes, these ribosomes are associated with the plasma membrane. sometimes. And even with mRNAs and form polyribosome. If you examine each ribosome consists of two subunits. One is smaller, other is bigger. Smaller subunit is called 30S subunit. Bigger subunit is called 50S subunit. S refers to Swedberg coefficient. ಅಂದರೆ ರಿಬೋಸೋಮ್ಸನ್ನು ನಾವು ಸೆಂಟ್ರಿಫ್ಯೂಜ್ ಮಾಡಿದಾಗ ಎಷ್ಟು ಬೇಗ ಇದು ಸೆಂಟ್ರಿಫ್ಯೂಜ್ ಆಗುತ್ತೆ ಅನ್ನೋದ್ರ ಮೇಲೆ ಸ್ವೆಡ್ಬರ್ಗ್ ಯೂನಿಟ್ಸ್ ಅಂತ ಮಾಡಿದ್ದೀವಿ. ಅದ್ರ ಮೇಲೆ ರಿಬೋಸೋಮ್ಸನ್ನು ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ. ಬಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ರಿಬೋಸೋಮ್ಸ್ ಆ್ಯಂಡ್ ಸೈನೋಫೇಷಿಯನ್ ರಿಬೋಸೋಮ್ಸ್ are one and the same. That is, they are of 70S ribosomes. Okay. Now you know the meaning of polyribosome or polysome. If many ribosomes attach to the mRNA, then it is called polyribosome. I already discussed the function. The main function of ribosome is protein synthesis. Okay. Next, last part, inclusion bodies. Inclusion bodies. The reserve food material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. The reserve material are nothing but inclusion bodies. These are not surrounded by any membrane and lie free in the cytoplasm. Example, phosphate granules, synophacin granules, glycogen granules, gas vacuoles found in blue-green algae and green photosynthetic bacteria, purple bacteria. Okay, so that's all about inclusion bodies and prokaryotic cells. Thank you. Thank you once again. So that's all today's class. Okay.